Some of the first part of this is really an overview. I'm a 30,000-foot guy. It's very hard for me to drill into talking about a receptor-ligand pair unless I know where it is and why it's there. So I'm going to give you a contemporary overview of innate immunity, how it blends to adaptive immunity, and then I'm going to end with some kind of a dangling participle about the microbiome and the evolution of the immune response, because it's very, very important right now. I will steer you to two books, and this slide is actually dated because the Immune System by Peter Parham has just come out with the fourth edition about 3 months ago. These are both by Garland Science, and obviously Janeway's Immunobiology is kind of the definitive version of this, but the Parham emphasizes human immunity and has less in animal models. It's a fabulous book, and I use that to teach my course back in Cleveland. So I want to start out, this is kind of a philosophy discussion. Why kind of like why are we here, but why is our immune system here? I want to answer that question. And I think our concept of that over the past 7 decades has changed. I want you to be able to recognize the integral components of innate immunity and have a clear definition in your head of what the difference is between innate and adaptive immunity. As part of that, I want you to be able to recognize the vital importance of these receptor-ligand pairs that are now part of the common parlance of how we generate an immune response. And then lastly, I'll talk to you a little bit about microbiome, and I'm going to recommend a book for you to read. So, why do we have an immune system? I mean, if we were sitting around drinking coffee here and I just asked everybody would have a different idea. Why do you have a circulatory system? What's your central nervous system? These are constructs. And now we recognize that most of this stuff is all cross-talking anyway. But the predominant answer, which I have got time after time, is that we need this to defend ourselves, primarily against infection. And I think that's certainly an important part of it, but hardly the whole picture. If we ask, in the history of medicine, which I teach in our medical school, when did the evolution of immunology as a discipline really move forward? I don't know. I mean, many people might go back to Jenner. Here you can see him on the left, immunizing to protect against smallpox. I mean, it's vaccination, this is immunization, this is a logical starting point. But at that time, this very small study, which only involved a couple handfuls of patients, there was actually no notion of a mechanism of action, and it was just protection. And in fact, Pasteur, a century later, who codified vaccinology, really had no scientific underpinning for what the basis of this anthrax or rabies vaccine might be. He actually postulated that, well, these organisms that I'm immunizing people with might compete for nutrients, and therefore another organism would not be able to take off. And it really was not till the very end of the 19th century that a couple people came to the fore. Emil von Behring, who won the first Nobel Prize in Physiology and Medicine, developed the whole system of serum therapy. And he knew that you could immunize a horse, and you could take the serum, you could protect people from this, and that was humoral immunity. And a very few years later, Elie Metchnikoff, who also won the Nobel Prize a few years later, postulated that it wasn't just this humoral mechanism, but there were cells, and this is phagocytosis. And these are fabulous notions to think about, but it gives us an underpinning. What I find interesting is that Metchnikoff, who was the father of this notion of cellular immunology, he spent the last 2 decades of his life interested in the microbiome. And he got everybody in the Western Europe to be eating yogurt because he thought that it's your bacteria in your gut is where all the health is. So this guy he was a pretty smart guy. So, all right. So infection. I guess that's a pretty good answer. But as you can get from my initial comments, you recognize that we swim in a pool of infection. And in fact, as we talk about microbiomics, you understand that we are outnumbered. We are not men or women. We are 100 to 1 microbes. And so the notion that the immune response is to protect us from all infections is oversimplistic. So there has to be a better answer for this. So the question is, how do we recognize those bacteria, viruses, fungi, that are harmful to us versus those that live with us and live on us, wet, dry microbiome? Well, the great advance, just to underpin our declarative knowledge here, is that by 1950, the notion that self versus non-self was important in the generation of an immune response was uplifted by the work of Sir Frank McFarlane Burnet, Australian immunologist who ultimately won the Nobel Prize. Peter Medawar, Argentine, did his work in Europe, also started noticing the relationship between tolerance and immunity in terms of skin grafting. So this was a very simple and very forthright theory that our immune system could tell what's us and what's not us. But it didn't really totally answer this because, we're always eating these yogurts and we're eating things and we're putting a lot of non-us in ourselves. We're covered with these microbes. There must be something different. Charles Janeway, the eponymous author of Janeway's Immunology, who died probably 20 years ago, made major contributions to our understanding of immunology, and he said, there's got to be something else. There has to be a trigger. Self versus non-self is a good answer, but there's good self, non-self, and there's bad non-self. And he postulated in an editorial written in the 70s called The Immunologist's Dirty Little Secret. And he knew that when you tried to immunize animals with a microbe, you didn't often get a very good response. But if you put in something which all of you probably remember from your undergraduate and graduate education called Freund's adjuvant, you could really boost this immune response. And it's just kind of this killed mycobacterium and oil and just a bunch of garbage. And he said, well, it must be doing something there. And what is it? And he postulated there probably were receptors for something in that adjuvant that revved up the immune response. And not only did he postulate this, and the reason that that we consider him reverentially in these discussions is that he went out and proved it. And he actually cloned one of these receptors. And that is the beginning of this notion. Well, further on from that, this is our latest hypothesis. And this is called the danger hypothesis. And I'm going to move forward, and I'm sure that 10 years from now this will be considered an oversimplistic reduction of this. But it's a way that I think about the integrated immune response. is that the immune system doesn't protect us from infection or non-infection, doesn't protect us from self or non-self. It protects us from danger, and that danger can be exogenous, like a pneumococcus, or it can be endogenous, like a urate crystal. And this theory was posed most recently by Polly Matzinger, who still has a laboratory at the NIH, and we will come back to define this. So Charles Janeway said these receptors which turn on the immune system, he called them pathogen-associated molecular patterns. In other words, our immune system could see these patterns on microbes. And we still use that notion. Matzinger said we recognize danger-associated membrane patterns, molecular patterns, and we're going to call these DAMPs. You want to turn on the immune system, your immune system will recognize the DAMP. So come back to that, recognize that notion. So the last time we did this course, I started with a story. And I said, you're walking on the park, and you got a nice sunny day, and you snag your finger on a rusty nail and generates this. So I'm changing the story this year. All right, so those good. Here's the tip. This is St. John's. Okay, so what do we know about going? It's pretty nice here, but this is a little bit different. So there's a great story, a fascinating story, and a horrible story going on in the Caribbean, and it relates around this mosquito. This is the Asian tiger mosquito. This is Aedes aegypti, and so many of you are nodding, you know where I'm going with this. So before 2012, there was no endogenous transmitted cases of the alpha virus infection, chikungunya, in the Western Hemisphere. And now most of you people are from South Florida, so everybody's hip on chikungunya, and all rheumatologists. It's like, gotta talk about this. But it's a great story. This is a virus, part of the alpha viruses, Old World, New World. The New World viruses are largely arthritogenic and are distinct because you get this viral infection, you develop some kind of arthritis, and they all have kind of crazy names and everything, Syndibus and Ong-Yong and Chikungunya. You used to think that's just what you did when you took a board test. In 2012, there were no cases in the Western Hemisphere, and now there are well over 1 million cases in the Western Hemisphere. This is not something that is theoretic. The virus is carried by a mosquito. It has an enzootic transmission pattern to remind us that it can exist in a host. The host is man. It is carried by the mosquito, and it can be transmitted to another man merely in that direction. It doesn't require an animal repository to create an epidemic. We'll track this over the next few talks of what happens to the immune response. The virus is transmitted by the mosquito. These viral particles have to generate an immune response, and they probably do it quite rapidly. The incubation phase is only a few days. And if you can't overcome this, the syndrome becomes ensconced, and patients develop constitutional symptoms and severe musculoskeletal symptoms that may last from many weeks to many months. So the immune system has to dance this choreography trying to get rid of the virus, protect the host, and return to immunologic homeostasis, and we're going to do that. Rheumatologists know this picture. It can look very much rheumatoid-like. This is a Syntascan and some episcleritis. This was sent to me several years ago by Professor Pandaya. Looks like a pretty serious form of arthritis, and now there have been local regional cases identified in South Florida, and we expect this to continue. So be aware. Now, if I go back up to 30,000 feet, this is the topography of the immune response. And I'll talk about it in three artificially separate areas. We'll talk about some anatomic and physiologic barriers. We'll talk about these areas of innate immunity, and then ultimately adaptive immunity, what we really think of in terms of immunology, or at least used to, T cells and B cells, and how they fit together. So this is a phylogenetic tree, and if you actually just look and see what's going on here, you can see this is all largely, prokaryotic, no membranes within the cell, and as we evolve ourselves up, here we are right here in vertebrates. Everything except for this little red spot, their defense mechanisms all utilize innate immune responses. So I will tell you that those cockroaches that have been around for 500 million years are doing quite nicely, and they don't have a T cell or a B cell. They have these what we used to be considered primitive defenses. I see Dr. Rigby over there. When he and I were starting out in immunology, no respecting postdoc was going to be studying anything other than T cells or B cells. You didn't want to study neutrophils or macrophages or stuff like that. Now it's totally the opposite, and that's the hipster area. So interesting. So this is my definition that I would like to have you recognize for innate immunity, that contains several components. Number one, this is our early defense mechanism, so early response. So that mosquito bites you, puts that chikungunya virus in, your immune response wants to get rid of it, and get rid of it now. And not only wants to get rid of it now, it wants to protect you from collateral damage. So early response, minutes to a few days. It does contain both cellular and humoral components, and we'll talk about them. Second point that is interesting, we think of memory as being synonymous with immunity, right? Everybody up to date on their adult vaccinations, and you want to get your vaccine because the next time that if you see a pneumococcus and you've had Pneumovax or you've had Zostavax or you're back up on your just basic vaccines, you'll generate a memory response and you won't get that infection. Innate immunity does not have that. Every time it reinvents itself. It doesn't add a memory response. And belying that notion is that we don't generate a more avid immune response in innate immunity. It is hardwired into our genome. So it's there. Turns on, turns off, turns on, turns off. Far more nuanced than that. But this is the definition. Those elements of the immune system that respond rapidly to harmful or dangerous stimuli. I'm getting that danger in there again, the DAMPs. By recognizing nonspecific and shared patterns, the DAMPs, occurring both externally on microbes and internally. And I've given you an example of urate crystal to the host. So let's move forward. Okay, so again, a generation ago, we recognized that there are anatomic barriers that participate in a very important fashion in our defenses. And everybody knows this. You have a burn patient. They're terribly susceptible to infection because their skin, their integument is profoundly destroyed. And in the gut, we have acid in our upper GI tract that obviously, with a pH of 2 or less, that can degrade microbes. So there was this notion, overly simplistic, that we were covered with some type of Teflon covering, and then when there was a rent in the Teflon covering, we became susceptible. Well, over the past 15 years in particular, it has become abundantly clear that these coverings are not Teflon at all. They are highly complex biologic systems that can defend us in a variety of ways. And these, a term that you should be recognizing, and I'll come back to this in my third talk again, are defensins. Defensins are these small molecules that are elaborated in the presence of danger that can actually, through electrostatic attraction and transmembrane disruption, kill microbes at the mucosal surface. This is very important in HIV host defenses, where the virus penetrates oral or anal or vaginal mucosa, and defensins are a strong line of protection. These have been classified and reclassified, and here is a recent review from Microbiology. Talks about that there are classification schemes that looks at their charge, and these are cationic defenses, and they are very important in host defense against a variety of tumors and a variety of infections. These also, in and of themselves, once they are stimulated by innate immunity, they can function as alarmins or DAMPs and trigger these DAMP receptors themselves. So think of this as a positive feedback loop. A danger signal is sensed by the innate immune system. These beta defensins and related proteins are launched in a kind of a Star Wars fashion that can attack the membrane, disrupt the membrane, and kill the organism. At the same time, they can light back onto cells of innate immunity and inflame the response by triggering these danger receptors. So I'll come back to the complexity of defensins when I'm talking about Th1 and Th2 again in an hour or so. So now let's just look at some major players. And the goal of this educationally is to have you recognize key cells of the adaptive immune system and be able to pair their primary functions in the integrated immune response. So here, if I show all of these cells, here's a hemopoietic stem cell, branching off into lymphoid cells and making, T cells in the thymus and B cells in the bone marrow. And here we have them differentiating into hemopoietic cells. Here we have this myeloid lineage that is prominently part of the innate immune system. But by the end of the day, you will be reminded of what you already know. These are not separate and distinct. These are highly linked, and we just kind of functionally talk about them separately. But there are more similarities than we have ever thought. All right, so the players. What should we be saying? These are dendritic cells. Dendritic cells, this type of dendritic cell, are offspring of hemopoietic stem cells. They come in two different lineages. One has differentiated along monocytoid pathways, the other on plasmacytoid. They are very potent scavengers of these dangerous stimuli, and they are the most important cell in priming an adaptive immune response. Many people have called them different things. Some people have called them wine tasters because they are sipping in the periphery at these foreign antigenic moieties, determining are you dangerous? Are you dangerous? Are you dangerous? Are you dangerous? If you're not dangerous, it's okay. I'll spit it back out. If it is dangerous, I'm gonna drink this, and I'm gonna process this and do this. Other people have called them sentinels. If you use a kind of a militaristic analogy for the immune response, these are located in peripheral tissues, in the submucosa, in the respiratory tract, under the skin. And as Langerhans cells, they will sample a signal, determine whether it's dangerous or not. And if it's dangerous, they do several things. First of all, some of these antigens will be taken from the cell surface. They'll be internalized. We'll talk a little bit about this. They're processed into little tiny bits and then presented on the surface in the context of histocompatibility antigens, looking for cognate T cells. The other thing to note is that once they have found these danger signals, they get a signal to travel. And that's very, very, very important. They just don't stay there. So if there are sentinels on the battlefield, they've identified a dangerous signal, and they're going to take this back. And where do they take it? They take it to regional lymph nodes, secondary lymphoid organs. And that's where command and control can occur, and they'll meet naive T cells to help turn them on. So the one thing that they don't do, unlike the next cell that we're going to talk about, is that they are not potent killers of these. And by design, we think. So when a dendritic cell finds a dangerous signal, it travels, but it doesn't kill. So just keep that as the most important part. And these cells, dendritic cells, are the cell within the professional antigen-presenting cell family that are most potent at activating these naive or virgin T cells. So we'll come back to that in a second. All right, second cell, macrophages. So if the dendritic cell is the scout, trying to determine if a foreigner is dangerous or not, what is the macrophage? The macrophage is a killing machine, and it has tremendous internal machinery to degrade dangerous external stimuli such as pathogens. Now at rest, they're not doing that. They're just kind of they're on day duty in the immune response. They're taking out the garbage. They're kind of keeping things in order, and they are doing that. They have house cleaning functions. But when they are turned on, and we'll talk about how they're turned on by cytokines, they then become very potent killers, and they can internalize and degrade pathogens. At the same time, they also are professional antigen-presenting cells because some of the antigens that they take in will ultimately resurface in the context of MHC, and they are capable of producing, stimulating an adaptive immune response. Unlike the dendritic cell, though, they're not quite as efficient because so much of the antigen that they've taken in, they are degrading and killing. So if the dendritic cells travel but they don't kill, macrophages are fixed in the tissue, but they're very, very potent killers. Now, the field of macrophage biology has exploded in the past several years, and these are cells of monocytic lineage, and in different areas, they have different functions. In the brain, we call them microglia, and they have a lot of housekeeping activities. In the lymph nodes, there are actually multiple families of macrophages. Those subcapsular macrophages are very, very potent antigen-presenting cells, and they're not doing a lot of killing. But if you look at the macrophages in the medulla, they are very, very potent killers and traditional. So whenever we see macrophages, whether they be in the joint, where cells of osteoclasts are of macrophage lineage, they can have very, very, very specific functions. Another way to look at these macrophages is the modes of activation. We think that macrophages are turned on largely by cytokines, Gamma interferon that we'll come back to time and time again is a very potent macrophage activator. Gamma interferon comes from activated T cells, as we'll see, and that's the traditional way. There are also alternative pathways of macrophage activation where immunomodulatory or immunosuppressive cytokines such as IL-10 may actually induce macrophages to engage in repair functions. So this choreography of macrophage activation is dependent upon the type of macrophage, where it resides and fixed in these tissues, and how these cells are turned on. So at some level here at this 30,000-foot discussion, yeah, there's a great deal of complexity, but understand that the role of these cells. Well, if they are the heavy artillery and killing machines, neutrophils are the foot soldiers. They are the ones that, do or die. They are constantly being produced in the bone marrow. They have very short half-lives measured in hours. These cells at one time were considered, boring. They come in and they phagocytose and they die. Now the field of neutrophil biology is romantic as dendritic cell or macrophage biology, and we know that while these cells have very short half-lives, the pathways through which they die have profound biologic implications. So I'll give, for the rheumatologists and dermatologists in the audience, this resonates easily. Normally, these cells are coming and going, and everything is quite safe. They die by programmed cell death that we now just know is recognized by the term apoptosis. This is how a cell implodes, and these nucleosomal ladders are packaged nicely. The garbage can be eliminated by an orderly process. This is happening all the time. We have no inflammation at rest, right? Patient comes into your office and they have these red spots that are slightly raised, and they have a fever and joint pains and hematuria, and you recognize these as inflammatory purpura or palpable purpura. And when you biopsy them, what do you see? Yes, leukocytoclastic vasculitis. And what that means in histopathologic terms is that the leukocytes have not imploded, they have exploded. And what you see there is nuclear debris in and throughout the biopsy. That's not good. And that has, in and of itself, is another form of death that generates a local regional inflammatory reaction. So those are easy to understand, these polar opposites. Well, now there is a third area, a burgeoning area in neutrophil biology that, perhaps next year we'll go into much greater detail. And this is a third pathway of dying, and that is known as netosis. When neutrophils die by netosis, the nuclear contents with DNA and their attendant histones and protein moieties flay out into the periphery in these structures that actually look by scanning EM like nets. They are capable of engulfing foreign material, and they in themselves are alarmins. Okay, I mean, an alarmin is a danger signal that has a danger-associated membrane pattern that can turn on the immune system. In certain diseases, such as ANCA-associated vasculitis, we now see myeloperoxidase within the neutrophilic nets. In rheumatoid arthritis, we now recognize that neutrophilic nets are enriched for citrullonated proteins. And these may be the stimuli in the story that Dr. Rigby is going to tell you later on about how we break tolerance and make antibodies to citrullinated proteins, so-called anti-CCP or ACPA, the basis of the molecular taxonomy of rheumatoid arthritis. So keep an eye, recognize the notion of neutrophilic nets, understand that they are part of our host defenses and are alarmins in the immune response. Finally, just a word that not all cells that make cytokines that are lymphoid in nature are T cells or B cells. This is a new category of cells known as innate lymphoid cells. They may have features of natural killer cells, or they may have features somewhere intermediate between them and T cells. They don't have T cell receptors, but they can be polarized just like cells of adaptive immunity to make cytokines of Th1, Th2, Th17, and all the things that we'll be talking about. So the goal of this in terms of your declarative learning is just to recognize there is a family of cells, innate lymphoid cells, that can produce cytokines similar to cells of T cell lineage. And this is why we've had such a hard time identifying these cells by flow cytometry, because there are so many cells that can do the same thing. So that's a high altitude view of cells of innate immunity. But this is also recognizing that these danger signals can be endogenous or exogenous. And here are some examples of these danger signals. Obviously, we see bacterial cell wall components, and we'll talk about like lipopolysaccharide. We've known about this for decades and decades. Microbial proteins such as flagellin or toxin from these things, we can get that. Nucleic acids. I've talked to you about, when neutrophils die by apoptosis, programmed cell death, they are not a danger signal. But when they explode, these degraded nucleoproteins can be danger signals, and within the context of neutrophilic nets. And then there are a whole family of other chemicals within the body which may be untimely. Uric acid is not a good thing. It can disrupt the integrity of phagocytic cells and lead to gout. There are some alarmins that are produced in the process of inflammation. Some of these have pretty crazy names, but the learning point is that danger can be exogenous or endogenous. So when we talk about these danger-associated membrane patterns, what really belies that? It's all well and good to understand this conceptually, but structurally, what's actually happening here? Well, there's a kind of a reductionist way of understanding it. Is that microbes, as opposed to humans, differ. We obviously differ biochemically. We have a lot of sialic acid. Microbes have much less, so that we have alternate glycosylation patterns of the outside of our cells. Within the microbe world, there is a lot of repetition. These highly simplified structures of surface glycoproteins that they in themselves can generate a danger signal. Here is a picture of mannose-binding lectin. Mannose-binding lectin is one of the three routes by which complement is activated. This is a molecule that looks very much like C1q with its collagenous limbs and globular heads. And what it recognizes here, this kind of lunar lander type of approach, is that on a microbe, it can bind to these repeating mannose units that it looks like a checkerboard. But on a human cell, while we may express mannose to a certain degree, they are sparse and asymmetric, and they don't allow tight binding, and thus we don't activate complement by binding to our own cells under physiologic conditions. So I think this is an interesting way to understand what DAMPs are. So there's a structural repetition that belies many of them that will allow them to turn on the immune system. Now, I will say this probably 10 times during these next 3 talks. If it were only that simple. You know, it's just like the behind the next curtain. And so, you know, damp receptors, this is a picture of a macrophage. This is from either Parham or Janeway. You recognize that if you have both of these books, the figures are the same in the books. There's just a few more in Janeway. And I really, really, really encourage you to get one of these because they are just lovely to review. So here's a picture of a macrophage, and it is literally festooned with an armamentarium of receptors that can trigger this innate immunity. Complement receptors, you know, yeah, why would it be binding complement? Well, because mannose could be binding to the surface of a bacterium. The lipid receptor, not all antigens are protein, and we can actually generate antibodies to lipids using some alternative antigen-presenting pathway. Scavenger receptor speaks for itself, and I'll come back to Dectin in a subsequent talk when we talk about defense against fungi. When these are triggered, these can be internalized and killed through these lysosomal pathways, and some of them may also have a gentler treatment through endosomal pathways and resurface to present to naive T cells. All right, so there is a separate family that everyone here recognizes this terminology by now of receptors in the innate immune system that is important that we take just a little segue to remind ourselves what the biologic role of these receptors are in immunity. I've shown you a bunch of receptors there, but now we're going to focus on toll-like receptors. This notion that this family of receptors, which has been known about for about the past 30 years, was discovered by Christian Nusslein-Volhard, who was looking at these mutated fruit flies that had a mutation in these leucine-rich proteins that ultimately were called toll. Toll in German means odd, far out, and when they saw that these mutated animals had dorsal ventral displacement of their wings and legs, it was just thought to be an oddity. If you look at this fruit fly, you see it's covered with fur, and that fur is fungi. They said, well, that's odd. So not only did they have twisted bodies, but they're infected with all this stuff. So it took a while later, in the next decade, Jules Hoffman, and then working on successive goals that had been reached, Bruce Beutler, all served to elucidate the function of these toll receptors. Nobel Prizes were all around. And we now have this system of TLRs. All right, so TLRs are part of our danger-associated receptors. There are 10 human receptors. They all have the same kind of leucine-rich structure. Some of them appear on the surface of the cell, and some of them appear within endosomal structures internally. So what are they doing there? Well, they're looking for danger. And they have the capacity to react to different things. So here's toll-like receptor 4, and it is canonically linked to lipopolysaccharide, though it can bind to many other danger receptors. Here are the ones that are internal on the endosomes. I mean, why are they internal? Why wouldn't they want to be on the outside of the cell, right? Well, no, they're reacting to dangerous signals that are internal, endogenous. Double-stranded RNA only can be dosed to a virus. Hypomethylated DNA, which is a microbial preferential pattern, et cetera. And then others, the flagellin and different types of glycoproteins. So these are very key elements in innate immunity. So what do? Here's what you remember. While the biochemistry is extremely complex and the pathways through which they generate their signal involve multiple linkers and adapter proteins, they basically do one of two things. They will lead to the translocation of NF-kappa B, a nuclear binding protein that is critical in all of these inflammatory pathways that leads to the synthesis and secretion of things like TNF and literally hundreds of proteins involved in inflammation. Alternatively, some of them also generate interferon. Okay? And we know interferon is an antiviral protein. So in biologic terms, when you trigger toll, you're generating an inflammatory response or you're generating an antiviral response. These are highly linked and far more complex than that, but this is a way to keep this packaged in your brain for remembering this. You know, many of the things that will be said during this day, which is trying to summarize an entire field of a discipline, these are like hand baggage for the mind. So TLRs, NF-kappa B, and interferon. Yes, it's a lot more to it than that. So if it were only that simple, we would be talking about this generation of TLRs and inflammation. But there are other families of receptors that have been elucidated. Here are the NOD-like receptors that you'll hear about tomorrow when we talk about inflammatory bowel disease, because there are gain-of-function and loss-of-function mutations in all of these that have contributed to human disease. Another family known as the RIGs. These are intracellular receptors that protect us against virus. Probably the receptor getting the most press in the past 6 months, STING receptors, which generate in response to viral proteins as well and are incriminant in immunodeficiencies and autoimmunity. Soluble receptors, pentraxins, such as CRP, which we use all the time, collectins and phycolins, these immune-based collagens, and then a variety of other receptors stuck on cells. So you're getting the picture that the innate immune system is well poised. It has multiple weapons to identify foreignness and to be able to tell us whether it's dangerous or not. Once triggered, a cell such as an activated macrophage then becomes an inflammatory machine. Not only is it potent as an intracellular killer and can function as an antigen-presenting cell, but it secretes a variety of cytokines that we'll come back to time and time again. These cytokines then act by recruiting cells and soluble products from other cells to actually defend the host. Some of them are chemokines, some of them create the milieu for generating T-cell effector function that we'll talk about in the next discussion. IL-6 coming from a variety of cells is the pivotal cytokine, but certainly not the only cytokine that contributes to this. And this often quoted paper by Irving Kushner from 1999 in the New England Journal really codified the notion that we talk about the acute phase response. Those proteins that increase by 25% during an inflammatory response. And this is actually being driven by cells of innate immunity that are releasing cytokines. So in the end of the day, what is the function of this innate immunity? Well, number one, it limits the extent of that initial danger signal, right? I mean, there's chikungunya. You've been bitten by the mosquito. That alpha virus has penetrated the skin. I'd like to get rid of it now before it has become disseminated. Unfortunately, that doesn't happen all the time. And if it's around for more than 3 or 4 days, it now needs to bring in the designer weapons. And that is cells of the adaptive immune response. So it primes the adaptive immune response. And then finally, whenever the battle is over, it has to participate in repair and tissue remodeling. So here we are. The virus has, you've been bitten 2 to 4 days, really not feeling too bad. But as you can see, this red line is the viral load, and within the first week of chikungunya, you have millions of particles per ml. And here you can see the green line. This is an interferon response. Where is interferon coming from? Probably from TLR activation. Here we're starting to see symptoms by 3 to 5 days, and the patient is now developing the severe arthritis, hence the name chikungunya from the Makonde dialect, meaning to bend or break. Very, I mean, in fact, anybody that's seen a case down here, you got to tell me about it. I've been looking for it. I just haven't seen it yet. And then, because this is persisting from weeks to months, we're now making antibodies, which means that we've had T cell and B cell cooperation, and we're expanding our T cell repertoire in response to this. And then hopefully, over time, the cells of adaptive immunity will downregulate and will maintain immunologic memory from antibodies in our adaptive immune response. And this is exactly what is happening. And if you look at a heat map of inflammatory markers in patients with chikungunya, it does not look very dissimilar to patients with rip-roaring rheumatoid arthritis. And you see TNF and IL-6 and IL-1 and gamma interferon being cranked out in these patients that are quite sick. So how has this happened? So now, at the segue here in the last few minutes, I'm going to just talk about, I'm going to segue to the microbiome, but innate immunity has run its course in the first 96 hours, right to here. It's been unsuccessful. The pathogen has escaped our defenses. That poor dendritic cell, that sentinel out there, he's got the info. He's got to take it back and has to kick up this immune response, and we're going to see how that happens in a few minutes. And that is the story of adaptive immunity. So let me just close here by just kind of this dangling participle of microbiome and the immune response. So as I said, I'll make an offer here to anybody that is interested. I have a monograph that I give to most of my patients. It's called What You Can Do to Maintain Your Immune System, a Healthy Immune System. And it talks about diet, exercise, and stress. And I have it as a PDF. It's all free. You can take it. It's about 25 pages. Patients love it. Because at the end of the day, no matter what we're doing for them, they ask you, what should I eat? You know, what can I do? This is, and it's scientifically based, but it's written, you know, for a person of a patient. So the microbiome and immunity. When we say microbiome, everybody knows by now. This is just the lexicon. The microbiome, coined term by Joshua Lederberg, is the sum total of all the organisms in our body. So, as I said, it's 100 to 1 non-human cells to human cells. So this is the notion that we are a community. We're not just us. And the things that we carry with us and drag with us are vitally important. Why? Because they are metabolizing nutrients. They're also involved in the generation of our innate and adaptive immunity, and that's why I'm ending with this. There's another term that you'll see in this literature known as nonobiotics, and that is rearing animals in germ-free environments and studying the effects of reintroducing bacteria and microbes into them. And then most importantly, this term metagenomics. This is the only reason that we're studying this. There's a huge complexity to this area, obviously. And the study of genetic material through advances in informatics have allowed us to do this. So yes, we have 100 trillion organisms in our body. 100 to 1 of them are of microbial origin. These microbes digest our food. They are involved in the development of innate and adaptive immunity, and they are vital to immunologic homeostasis, vital. These are the two advances that summarize why we talk about this. There are genetic probes that we call 16S ribosomal sequencing. All of these microbes carry a very conserved ribosome that allows you to merely sequence all the genes within the microbiome within the bowel, and then come up with a taxa of different phylum, class, order, family, and actually genus. That tells you what kind of organisms you're carrying, but it doesn't tell you what they're doing. And then more advanced sequencing techniques have allowed you to look at all the genes expressed by these, and they can be ordered in terms of their function. Now, this sounds so easy. I heard a microbiomic guy from Stanford say, it's like going to a desert island and having a massive chipper shredder go through the entire island and reduce it to a pile of rubble. And then someone comes along and takes a paper bag and puts a scoop of it in here, and then hands it to you and say, tell me what this island looked like. And that's what it is when we sample the microbiome within the bowel, or a navel, or an ear, or a gum. It is very challenging. So these highly reductionist comments about, well, take this probiotic and everything will be okay, is probably a kind of a reductionist view of it. Nonetheless, this is very exciting material. So this summary figure from a recent Nature Immunology review reminds us that our bowel, we are born sterile, and then rapidly acquire this expanded microbiome. And that is highly influential in the development of our immune system. If you look at animals, these nonobiotic animals, they have defects in integrated host defenses that are modified by reintroducing microbes into the host. That can be done in the right way, or it can be done in the wrong way. And this notion of dysbiosis, we are infected with wrong microbes, may belie some forms of autoimmune, autoinflammatory, or immunodeficiency diseases. There are notions that if you take a mouse that's nonobiotic and then reconstitute it with the fecal matter of a human, those mice will be immunodeficient in many ways. But if you reconstitute them with the fecal matter of a mouse, they'll regenerate their own robust immune system. So at the extremes, it's not just having bacteria. There's a texture here, and it may be the right bacteria. This is important, and there are some experiments that I think are very straightforward examples of the importance of this. So If you take conventionally housed mice, that means that they have bacteria all over the place, and you expose them to CMV, they will deal with this much more efficiently than mice that have been previously given high doses of antibiotics. So in other words, they've had their bowel partially sterilized. So we think about this epidemic of antimicrobial use that we do. What are we doing? Are there any downsides for this? Well, possibly. Antibiotic-treated mice succumb to sublethal amounts of influenza as opposed to their conventionally housed litter mates. So again, antibiotics can have an effect on this. And then peritoneal macrophages from antibiotic-treated mice, if you look and actually ask what the molecular pathways are, there are evidence that some of these receptors, remember the RIGs, not the tolls, not the nods, but the RIGs, they're intracellular sensors of danger, are downregulated, and they make defective interferon responses. So the question is, can we do anything about this for ourselves by enriching our body with the appropriate microbial spectrum? You know, I'm going to close by just giving some philosophy here. I mean, actually, the whole talk is philosophy. So, but this is interesting. So we've existed, you know, for eons. The human species developed millions of years ago, and the genetic drift of our species is pretty darn small. Less than 0.5%, according to those people that can figure this stuff out, per every million years. So over the course of a few million years, our genes have changed just a tiny, tiny bit. On the other hand, our environment and our diets have changed in a frame shift way over a few centuries. So the question has been raised by, you know, philosopher immunologists, is that have our bodies been able to keep up with the changes to our system? And I told you, we are a community. I've made the point here. I think everybody can buy into that. These observations, this is a famous paper that's been quoted many, many times over the years from a few years ago that have epidemiologically tracked infections over the past generation. And rheumatic fever, tuberculosis, measles, mumps, all the things that we protect ourselves have gone down, down, and down. Over here is the pattern of autoimmune disease going up, up, and up. Now, I'm going to stop everybody right now. You're saying, well, this is some epidemiologic bias, this, that, and the other thing. It is not epidemiologic bias. It's a complicated thing, but it's incontrovertible people that study the epidemiology of autoinflammatory and allergic diseases that it has been on the rise, and it's not just recognition and diagnosis. So what could belie that? So the last thing that I'll tell you is that I want you to read this book. This is a great book. This is called An Epidemic of Absence. I have no conflicts of interest with Mr. Velasquez-Manoff, but I'd like to invite him to Cleveland, actually. And this is a part of a book review by Abigail Zuger in the New York Times, who's so thoughtful about this. It says, even the most critical reader has to give Mr. Velasquez-Manoff credit for the prodigious task he has undertaken as he shovels around the reams of data, most of which he lacks the expertise to interpret. But when I went to this book, I went to the back, he has interviewed virtually every major immunologic heavyweight on the planet. And he has alopecia universalis. He has no hair. And he's been looking for a natural cure for this. So it starts out that he's going to Mexico to be infected with hookworms. He actually doesn't do it, but it's a story. It's really, really a highly scientific and very good book. So his postulation is that, and according to many other people, is that over the past 100 years, we've gone from everybody harboring parasites to nobody harboring parasites, at least in the West. And that parasites had shifted our innate and adaptive immune responses and have skewed it to an allergic and autoimmune profile. It's way beyond what we're talking about here, but I'll be happy to chit-chat with anybody on the break. So I'm going to stop here for a minute, open this up for any kind of questions, editorial comments. I see Dr. Silverman in the back who could wax poetic on this microbiomic world. Dr. Rigby's over here. So thank you very much. I'll stop here.